Ready?
So, good afternoon to all. This is the Mosqlimate seminar. We are in our sixth edition. Today, our invited is Paulo Ventura, a postdoc fellow at Indiana University. Welcome, Paulo. It's a pleasure that we are with us today. Thank you all for being here to listen to Paulo. And please, Paulo, share with us your presentation, please.
Oh,
And
all right. Thank you. It's my pleasure for sure. Wait, then can you see an email or the presentation?
the email.
Okay, so that's wrong. Let's
Mhm.
start over and desktop one. Okay, now you should be seeing a blue slide, right?
Okay, Paulo, we usually have a meeting of an hour. So
Mm-hmm.
if you can talk in 40 to 50 minutes, we are okay
Okay, perfect.
with
Yes.
presentation.
Okay, thank you. So yeah, I think it'll be a relatively short presentation. I didn't have a presentation ready for this work, so I made the slides. Well, first of all, thank you very much for inviting me. It's really a pleasure to be able to show my work to you guys. I have been checking the Mosqlimate platform for quite a while now, so it's really an honor. And let's start talking. My name is Paulo Ventura. I am currently a postdoc at Indiana University, School of Public Health in Bloomington. And well, let me just talk a bit about me. So I have a PhD in physics. I got it in University of São Paulo, in São Carlos. During my PhD, I did an internship at Universidade de Zaragoza with Professor Yamil Moreno. And after my PhD, I got this position here as a postdoc at Indiana University. So, just for some context, in my PhD, I was working with epidemic modeling, more like on complex systems and the I I view of like complex systems and on complex networks. But then here I got this job and we are focusing on applied questions and like models to answer applied public health questions. So without further ado, let's go for an introduction. And I think I don't have to spend much time here explaining the importance of dengue, the burden it causes. Indeed, it's the highest burden among all arbovirus diseases. And it had a steady global increase, let's say, through the past few decades. I'm specifying these dates here because there's a reference, but it has a relatively steady but increasing trend. But recently, over the world, around the world, and in Brazil, we had some spikes, like in the last few years, and it seems like there's some sort of shifting pattern. Things are like starting to spread quicker and for new places. And we do associate this with climate change as a major driver, often also about we related to urbanization, oscillations of climate like the El Niño, population growth, etc. However, the interplay between the pathogen, which are four dengue serotypes, DEM serotypes, host immunity, which is not so simple with respect to those serotypes, the mosquito vectors and their relationship to the environment is a complex interplay. So there's a lot in the table on when it regards to dengue dynamics, and so there might be more factors there. And predicting dengue in the short, mid, and long term is a challenging. Essentially, from the modeling perspective, is not the easiest of the diseases to model because of all this complex interaction between many factors. So we have questions that I think they're very relevant, which are like, will these trends keep going up? Are we gonna continue seeing dengue increasing and increasing in incidence and its overall spread on the world? And more importantly, can we combine some types of interventions to revert this growth trends? Because so far we're seeing it increasing despite some degree of interventions already in place, right? So, and by the way, like I said, it's a short presentation, so I invite you to interrupt me and ask questions in case you feel like doing so. So feel free. And well, for some of these questions, mathematical models can be useful for addressing them. And here it's more of a me classification of models, but I like to see models as starting from a minimal models spectrum, like that we use for more theoretical analysis, understanding the theory behind that dynamic system, how chaotic it is, what is causing this chaotic behavior. We can have ingredient-rich models in which we try to incorporate many different ingredients from reality and see how they play along. So that's very nice to study the effect of these mechanisms. And we have what I call predictive models that I like to see more of like in an in-between between being minimal and feature-rich, because we want to have the necessary and sufficient ingredients to explain real data as best as we can, right? So we have to find a balance here between many ingredients and minimalism. And we're gonna be on this side of the spectrum here. So essentially here at Indiana University, we work with predictive models. So every model we work is grounded on data, and we want to use those models to address practical questions from public health authorities. Okay, so that's essentially our scope here in the spectrum of mathematical models. So for this presentation, I'm going to focus on one of the works that we already published, which is a short-term forecasting for the Aedes aegypti vector. And it's published in PLOS Neglected Tropical Diseases. But then I'm going to do an overview of other works and then some future directions that I am considering. And okay, so let's introduce this work. So what's the problem? Is that local mosquito control authorities in cities and counties, they require more tools to provide them situational awareness. So, and by situational awareness, we mean like consciousness about what is the current situation in terms of like the epidemiology of the disease. So nowcasting, short-term forecasting, estimation of quantitative metrics like the reproduction number or categorical incidence, pretty much a lot of what InfoDengue already does in Brazil. So authorities do want this. And this study is going to be focused on cities in the US. So the literature for forecasting specifically, which is what we do, is extensive indeed. It's not like it's a new thing, but I noticed that it emphasizes dengue incidence rather than the abundance of mosquito vectors. So we can see both, but usually you find a lot more papers and forecasting the disease incidence. And why do we care about forecasting mosquito vector abundance? Because, well, that provides awareness of the risk of transmission, right? Because when you have a bigger pool of mosquitoes, you have a higher risk of being infected the dengue virus is introduced, right? And well, speaking of cities in the US, overall, dengue is not endemic in the USA, continental USA, I mean. But vector species like Aedes aegypti, which is the main vector for dengue, they are established. So there is an endemic population of the vector there. So what happens, for example, in Miami is that from time to time you have local transmission outbreaks of dengue, also Zika, but they usually start and end with leaving like no further traces of sustained transmission. So for these locations, forecasting the abundance of vectors is also a way to measure what is the risk of an outbreak in case dengue is introduced in that location. Okay. So our objective for this work was to develop a short-term forecasting tool, where short-term here I mean 1 to 4 weeks ahead, for the relative abundance of Aedes aegypti in urban areas of the US. And we achieve this through a methodology that's based on a mechanistic slash statistical method. I'm going to explain this better. But it's interesting because having a mechanistic component, it provides more than just a forecast. It provides some mechanistic understanding of the system being studied. Okay, so let's go to describe the methods. This is a summarized scheme, but I'm gonna try to explain on a brief and linear way. So first of all, the data set we have is a data set for adult female aegypti mosquitoes collected in CO2 baited traps. And this is for 4 locations in the US. It's Los Angeles County, Key West, which is here, like in the Florida Keys, Maricopa, which is the county where Phoenix, Arizona is, so this city here, and Miami, and Miami-Dade County. Okay, so, and one thing that we did is that we normalized the total collection of these adult female aegypti mosquitoes by the collection effort. So we are trying to actually measure the abundance, trying to reduce the bias due to the collection effort. If you see those orange lines here, this is how many traps were deployed each week. So some locations are more stable, some are not. So we just try to minimize the bias by dividing by this quantity. So that is the quantity of interest. And the model itself, we can state it is a renewal equation model. So renewal equation, we might also know it as the Kermack-McKendrick model for infectious diseases. And the deterministic form has this very simple equation in which the incidence of the quantity of interest at time t, so let's say today, is a consequence of the incidence you had in the past. So at time t minus s, and is driven by the reproduction number here. So how many secondary cases a primary case generates, and the distribution of the generation time. So which is what is the delay between a primary and a secondary infection. And this is nice. It'll give you a number in the end, but that's deterministic. But since we're talking about probabilistic forecasting, we might want to have a stochastic version here. And so we just take this average and assume that it's just driving a simple Poisson process. So the renewal equation for us is a Poisson of this, and then the actual incidence is a random variable. Okay, so far? Okay, but this is more well known for forecasting infectious diseases. But here was, I said I'm going to forecast the abundance of mosquitoes. So we have to adapt a little bit the concepts here. So essentially X here for us will be the abundance. So the normalized number of specimens that are collected. This is the reproduction number, but we might actually want to interpret as the average number of surviving offspring. So think of like you have a parent mosquito and it might generate some eggs if it succeeds to mate. Then some of those eggs will die in the process. And some mosquitoes will make it to the next generation and generate new eggs. So the reproduction number here is related to how many mosquitoes effectively go through an entire generation in average. Okay, and the generation time is going to be the time between, again, surviving generations. So we, again, concerning about surviving mosquitoes between one and second generations. And this is what will drive our renewal equation here. Okay. All right. So in a nutshell, the method can be described by this slide is that we start from, let's say, the relative abundance in weeks, and weekly resolution, sorry. Then we estimate the reproduction number RT and the generation time from the past data. So we're estimating past R and phi here. Then we forecast the reproduction number and phi into the future, where by future I mean 4 weeks ahead, with the method that I'm going to explain. And then having R, having phi, and the past incidence, we use the renewal equation to forecast the abundance, essentially getting the forecasted time series. And it's stochastic, so we'll also have some confidence intervals for that estimate. Okay, so that's very roughly summary of the method, but let's dive a bit more into the steps. So to estimate the past generation time, we're using a compartmental model here. So it starts from temperature data and use some temperature-dependent mosquito development and mortality parameters. So we go through the simulation of eggs, going to larvae, then to pupa, then to adults, which will lay eggs. The only modification to your classical compartmental model here is that those eggs will not continue in the simulation. We just want to count them so that we also want to check the delay between parent and children And essentially that is the generation time. So that's how we estimate, and we get this over time. Also, the generation time distribution is fit to a gamma distribution so that we can better use it in the next steps. Okay, then the reproduction number. There are many estimation methods, but we chose a simple one that is very closely related to the renewal equation. So we start from the past data, the abundance. We preprocess it, and I'm going to explain briefly later how to preprocess. And then from this, we essentially invert the renewal equation using Bayesian approach to infer what was the reproduction number that explains the observed abundance. So from the renewal equation, if we invert, we have that R of t is a gamma distribution that depends on the incidence and on the generation time calculated in the previous step. Okay. Okay, and the preprocessing, it's important to mention because essentially it's what will give us free parameters for the calibration. So we do a low pass filter that removes the noise, and what I call noise here is like short-term oscillations or fluctuations. This type of data has a lot of short-term fluctuations. And we interpolate it from weekly to daily because week is a bit of a coarse time scale for the forecasting. And then we scale it up, and then we fix negative values that might have been generated in the interpolation process. Okay, so this creates a smoother time series that is better to work on. And then the fluctuations might be later captured by the confidence interval. Okay, so how do we forecast then? Essentially, we had the past reproduction number, so we forecast first the reproduction number based on the historical trend and the recent past. So essentially, we take an average of the recent estimates of the reproduction number and we multiply, but the historical ratio, so how historically you see this reproduction number changing. So we start from now and use historical trends to change this reproduction number over time. It's a simple forecast. We tested a few methods. This one seemed like the most promising. And for the generation time, we do something even simpler. We just take the average over the last 7 days, and that is our future generation time. Reason why is that, again, that was the simplest and already enough working method for us in our analysis. So we have future generation time, future reproduction number, and we have past abundance. So these ingredients, we plug them in the renewal equation, and it allows us to estimate the future abundance, which is x of t here. So that is simplified. There are lots of other steps, but this is essentially how it works. We estimate past reproduction number, generation time, project them into the future, and then use these to reconstruct the future abundance, okay? Okay, so let's jump to the results. So starting with something more visually easy to interpret. So we have here the data for the 4 locations that we studied. And then this is the darker line here is the data, and our forecast is the lighter shade of blue that you see here as the trace line is the median of our predictions, and the shaded area is the 90% confidence interval. And what we see in general by a visual inspection is that overall the forecast is doing well. So we see that it follows the data, it more or less captures some oscillations, right? But if you look at some seasons, for example, this one, it was a bit late. It didn't immediately capture that there was an increasing trend there. Or in the case of this very outlying season in Maricopa, it was also late, right? Eventually, it tried to follow the data, but that took a few weeks of it to realize. But the good thing is that it's following the data. So this is a situational forecast. It's really taking into account the recent past. And okay, but that's nice. It's visually simple to interpret this. And sorry, this is the 2-week ahead forecast. So for each point, you're seeing here what we predicted 2 weeks before, essentially. Okay, so now let's quantify a bit better the performance. So these are summary of the performance of the forecast. So starting with the left panel, this I'm gonna call a dispersion of points here. Essentially, y-axis is what we predicted as the median forecast, and in the x-axis is what was actually observed. And we want these points to be ideally close to the identity line, okay? And overall, they are at least around the identity line, so there's not any bias towards like higher values or lower values. The dispersion is like around the center line. Then for an accuracy metric given by an error score, this is the SMAPI score. It's essentially calculating how far the data was from the prediction in the median still. We see that by locations, we had some locations that are Los Angeles County and Miami-Dade County. They had a better performance, possibly because Los Angeles and Miami had a more like regular dynamics, other than, for example, Maricopa that has this outlying season, or Key West has a more fluctuating overall dynamic. So in these two locations, our model did better. And for the coverage of the confidence interval, and this is calculating essentially for each confidence interval, the 20%, 30%, 50%, how many of the points were inside this confidence interval. What we want to see here is something close to the diagonal line, because if your model promises 50% confidence in this range, you want more or less 50% of those points to be there. And overall, we see that the coverage was good. Maybe clustering by locations, we see some undercovering. So maybe due to some outlying points that are not falling inside the confidence interval, but overall, it is there. Okay. Okay, so let's look at the same performance metrics, but for grouped by other variables. So here we're seeing them grouped by horizon. So 1 week, 2 weeks, 3 weeks, and 4 weeks ahead of the forecast date. And for the dispersion graph, there's not much to interpret here. But when you look at the SMAPI score, the error score, you see a clear pattern that earlier weeks have a better performance, because lower here means better. And that makes a lot of sense. We're usually better at predicting what happens next than what happens farther in the future. So that makes sense. And for the coverage here, the good thing to say is that the model didn't start to overcover or undercover. So for later weeks, maybe the confidence interval was a bit higher than the ideal, but it did not have any great big deviation from what is expected. And now with respect to mosquito activity, so aegypti activity, and we had a way to classify between low, medium, and high. It's related to the smoothed version of the data, and then we classify with respect to the peak of the season. But essentially, in points where the activity was low, so again, there's not much to discuss from this figure, but you see some outlier points that are mostly for low activity. And here there's another clear pattern. So for low activity, the model was not doing as well as for medium and high activity. In fact, so if you have low activity, you have less data to forecast. So it's also understandable that the model is doing better for higher activity. And here we also see that the coverage of the confidence intervals is higher when there's a high activity, other than when there is a low activity. Okay, so, and fine, this is the method itself and how it does. But how does it compare to, let's say, a naive baseline approach? So is it doing any better than some unknown, some like silly guess, let's say? And we use a naive like baseline approach that is adapted from one that's used for the CDC forecasting of respiratory diseases. And essentially, it does a flat median. So you start from the point today and you predict a flat evolution. And the confidence intervals are calculated based on past differences in the data. So you take the differences and sample from those differences, and that's generating your confidence interval. Well, the green cells here are the circumstances where our model did better than the naive. And fortunately, we see that it did better in most circumstances. So for most locations, the WIS score didn't explain here, but this is a score that measures both the accuracy and the width of your predictions. But it did better than the naive in all locations, with Key West being borderline. So 1 here would mean same performance. Smape is fine, but Maricopa didn't do good, but maybe because of that outlying season. Coverage was always better than the naive. And also for the horizons, it's fine, except for one metric in the 4 weeks ahead, which is okay. And low activity, yeah, the model consistently did not do that well, which again is fine because we have less data to forecast. And what is more important is that we forecast in when there is activity, right? We don't mind much forecasting the off season. So the medium and high activity are the most important when things are happening. Which takes me to the conclusions and discussion. So we developed a relatively simple predictive model for aegypti abundance. And the interesting thing is that this model also involves estimates of the generation between mosquito parent and children and the reproduction rate. So it's not just a forecast method, it's a mechanistic approach that provides you some extra knowledge in the system rather than being just a black box that does the job. Um, well, the performance was consistent with overall superior accuracy over a baseline model. And the confidence interval were providing the appropriate coverage. So meaning that those diagonal lines here, they were sticking to the diagonal, meaning that, yes, the model delivers what it promises with respect to confidence intervals. And this forecast can provide situational awareness to public health authorities. So this work was developed in partnership with the authorities of Miami-Dade County, Maricopa County, and then Los Angeles County. Key West is a public data set, so we just fetched it online. And yeah, so there is a demand for this. It's currently not in a public interface or something. We just tested the model, but it is something that is on demand. Right. Okay, so like I said, I'm gonna skim through some other works that we do here. Really just 1 or 2 slides each one. So first, we have a few works, a series of works regarding mosquito diel activity. And diel activity means patterns of activity during a day. So let's look at the left side figure here. If you look at the x-axis, you see the hours of the day. And this is the result of a surveillance by hour. So, for example, this is how many Aedes aegypti mosquitoes were collected by hour of the day. So that's a very particular surveillance that these two cities provide, in which you don't only deploy the trap for an hour and aggregate it over days. So, and that allows you to see like the preferences for activity of each species. There's clearly much more aegypti and quinquefasciatus in these locations, but you see that they have different habits, right? So aegypti is more active here between 8 and 9 a.m. and then 6, 7 p.m., but Culex quinquefasciatus seem to enjoy more the nighttime, right? And one important note is that this is CO2 beta traps. So these are female mosquitoes that were probably looking for blood meals because the CO2 is what we emit and it's what they use to go look for humans to bite, right? And a similar thing here for Miami-Dade County. But why do we care about this deal activity? Well, because it helps you improving the performance of insecticiding application. So usually authorities will just spray the insecticides when they can, but if the mosquitoes are not out there, maybe they won't be reached by the insecticide. So I'm thinking here about the truck spraying, where mosquitoes, if they are just hiding, they will not be found by the poison, and so they will not be killed. So based on that and on a compartmental model, the team calculated essentially how much you can reduce the number of infections by dengue or by dengue, depending on the hour when you apply the insecticiding. And it's interesting that there is a clear pattern here. So the baseline has no interventions, and depending on the hour of the day, you can essentially cut the outbreak or well, sure, have some effectiveness, but not that much. So it's an interesting question and it's relevant, both for mosquito control and then for the effectiveness in curbing dengue transmission. Um, and then, when this is a work that I first authored, we can combine this mosquito deal activity with human deal activity, essentially. And by human deal activity here, I mean especially outdoor exposure. An important note is that this is, again, for cities in the US, where buildings are essentially closed and are conditioned, like the vast majority. So mosquitoes don't usually enter buildings, and humans are really exposed when they are outdoors. So we use the time use data set to know what activities people are doing, and then we classify those activities between most likely outdoors and most likely indoors. And this is the kind of pattern that we see, right? So clearly, humans usually sleep at night, work at day. And the interesting thing here is to combine these two activities to find out when humans are actually more exposed, right? And so this curve here is a combination of these two by a simple multiplication. And it reveals us that for aegypti specifically, uh, there are some hours in which humans in the US will be most exposed by this species. So by bites of the Aedes aegypti, right? And it's interesting because sure, the peaks are more or less the same peaks as of the activity of the mosquito, but here you see that this peak here is higher in mosquito activity, but it's not the highest in availability of biting, essentially, right? And this can be then used for also simulating dengue dynamics, but we didn't get to this point yet. Yes, and there is another work. This one I'm just showing a conceptual figure because we haven't submitted anything yet, but we have results already. Just waiting to finish the manuscript. But here the idea is to make a in silico trial of an intervention. And for this case, we specifically focus on the genetically modified mosquitoes that there are a few techniques, but essentially we focus on one that you release genetically modified mosquitoes in the male mosquitoes in the wild, and they will mate with wild type females, and the offspring will carry a gene that makes the female unviable. So these females will not participate, they will not generate offspring. And what is interesting about this intervention is that the offspring, the male offspring, still carry one allele of that gene. So they are heterozygous, and they will pass this gene to at least a fraction of their offspring, and so on and so forth. So that is an intervention that is interesting. Few trials have returned some mixed results. And so we wanted to develop an in silico trial, right? So it's a trial based on computer simulations for calibrated models. So the models are calibrated with real data. And with these, we provide a guideline like what should you expect on a field trial of this intervention, given these parameters, if you start on this date of the year, if you release that many mosquitoes. So that should be a guideline for people that are applying this method in the field. Okay. And well, so that was current good works, ongoing works, but I just wanted to briefly skim over a future direction that I particularly am trying to start with, which is I call the inframine dengue. It's the in silico framework for modeling infectious diseases. So this is essentially the name that I gave to a project that I am writing. So, and essentially I want to start writing simulations for data in Brazil. So I'm going to show this figure. I think it's an overview, but essentially I want to develop this family of formulations. Um, and so by family of formulations, it means I'm going to have a renewal formulation like the one I presented, compartmental formulation, which is more like a base for like developing compartmental models, and then an agent-based models for the most demanding needs. Like if you need a lot of details, it's a lot easier to use agent-based. So we want to have them three all under the same umbrella for like code standards, an interface, documented code. So hopefully to have this software infrastructure to facilitate development and future use. And then also physical infrastructure that I'm applying this to the Physics Institute at Unicamp, And so they have a cluster, so we'll be able to use clusters. I'll also quote, I'm also putting in the budget and workstation in which we can run the simulations without waiting in queues for clusters. And so the idea is to have all this established infrastructure for modeling so that we can quickly prepare models when we need. And well, the main data stream that I am putting on this project is the data that is available on Mosqlimate. So we have dengue incidents from InfoDengue. It's already now casted, which is great. We have the abundance now from the Conta-Ovos system, which is more recent, but it's also essential if we want to calibrate models that have the explicit vector dynamics. And we also have an interface for meteorological variables. So we have other data sets on the literature, but it's nice that Mosqlimate has a unified, standardized data set. So it's going to be the main data stream. And with this, we want to calibrate models for locations where enough data is available and then produce predictions, both for like a short-term forecast, like I just described, a midterm that I call here, and that's more a seasonal outlook. So outlook for one season, and then some long-term trends. So I know that it's hard to predict things for the long term, but maybe we can try to see what are the trends, putting some climate change projections. So this should be seen as an open question. Uh, and especially the midterm predictions, it's something that I plan to submit for future editions of the Mosqlimate Sprint, if hopefully we continue having it. So, and I also want to use this predictive framework for testing interventions, and I want to focus on combinations of interventions because there is some literature on interventions alone, but I believe that combining interventions is the key to possibly reducing and curbing the incidence of dengue. So I think that's it for me. So I'd like to thank you all for the attention. Thank you again, the Mosqlimate team for inviting me. Again, it's a honor, and I'm very open to question and discussions. Thank you.
Thank you very much, Paulo, our invited speaker. It's a fantastic talk, really
Thank you.
wonderful.
Thank
I
you.
would like to, well, if someone have some questions, Professor Flavio, if you want to talk.
Well, maybe somebody else will want to start, and if there's some time at the end, I'll ask also.
I'll see the chat.
Do you raise hands? I don't see the raising hand option.
Well, if you want to
You
ask your
can
question,
begin. Mhm.
yep, go ahead.
No need? Okay. Okay, so my question is about the baseline that you use for your model evaluation. So you
Okay.
used a baseline, pretty much just the current average, right?
Mm-hmm.
Did you try something like did you compare? I know it's not in the
Mm-hmm.
work here in the presentation, but did you
Mm-hmm.
compare with like perhaps an ARIMA or something else like that is a little bit more sophisticated,
Mm-hmm.
but it's also very simple. I mean, you just need the raw data for the time series. You don't need much else, and you can already
Mm-hmm.
get perhaps a little bit better results, getting some trends, or no?
Yeah. Okay, so thank you. It's actually a great question because we are trying this now. It's a very early process. We're still like engaging some students in implementing those models. But yes, we do want to do that because sure, it's nice, it's mechanistic, but it's also tied to the limitations of such. And maybe some statistical model, whether it's simple or not so simple, could do just as good or even better. Who knows? And maybe we can then try an ensemble of such methods and see if the ensemble itself does better. So these are, I think, definitely future directions. And it's something that if I have the chance for this project that I'm writing a grant with APESP, I definitely want to try this. It's actually part of my proposal. And I'm welcome to also suggestions and collaboration ideas.
Well, perhaps I'll ask my question now. So, yeah, well, first of all, Paulo, thank you for the great presentation, and I'm really happy to see that you're using our stuff, and that's why exactly that we built it. So yeah, so it's always a pleasure to see other people making use of it in such a very interesting project and some elegant approaches. So yeah, I'm really happy. But I have one question about, I mean, you didn't talk about fully about the full integrated from mosquitoes to dengue, even though I know that your
Mhm.
group works on that. But there's one question that is always on my mind when we talk about fluctuations of mosquito populations and its consequences in dengue. And I'll try to describe it very simply. Sometimes when you have a forcing, like if you think of the mosquito population fluctuations as a forcing functions on the transmission of dengue,
Mhm.
and the general idea, the first thing that comes to people's mind is that, well, okay, this is pushing the system across some sort of threshold that creates an that pushes the R naught of dengue
Mhm.
above 1 and then below 1. But one thing that I think is true for many locations is that these fluctuations in mosquito population is so above the minimum requirements for transmission, and that's just a personal view. It's not there's
Mhm.
It's very hard to measure that.
Okay.
That it's not exactly the fluctuation
Mhm.
of mosquito population that is driving the R naught for dengue above and below 1, or the RT,
Okay.
if you will, but something else, something related to that. And we've done some work
Mhm.
on these ideas. So I don't want to extend myself. I just want to hear your opinion on this.
Okay, yeah. So sorry, let me just see if I can understand. So we're talking about those short-term fluctuations in the data. Maybe
Yeah.
if I get back. Okay. Yeah, so just ask me again if I don't understand exactly what I should be discussing, but essentially what are the drivers of these fluctuations, or how, or whether it drives dengue or dengue transmission, right? Okay.
Yeah.
Okay. Well, I gotta be honest, I spent more time with myself thinking about the first question I just posed, which is like, what is driving those fluctuations? Because for me, it's very puzzling. It looks like any random events or random circumstances around the traps can cause huge differences. And I want to try to investigate the correlation with this and some meteorological variable, but I think it's challenging. Now, for the fluctuations and its in fact effect on transmission of dengue, I have not thought about this and what is causing. So when you say fluctuations in dengue incidents, do you mean like short term as well, or more like
No,
between?
no. I mean the main epidemic season,
Mm-hmm.
because you see that as climate drives mosquitoes, climate
Mm-hmm.
also drives dengue,
Mm-hmm.
but climate doesn't drive dengue directly. So it has to drive it through mosquitoes.
Mm-hmm.
So the mechanisms that people think about how this operate is, I think, maybe. There are some possibilities, and it's not
Okay.
a well understood thing.
I see. Okay. Looks like we
I
have
mean,
an
it's just a
interesting
provocation.
question.
I
Yeah.
mean, it's an open question, because
Uh-huh.
what I was trying to say is that there's so many mosquitoes. It doesn't matter if they fluctuate or if they stay constant in terms of what happens to dengue by the sheer
Okay.
number of mosquitoes. But we have a paper for a few years ago where we look at the extrinsic incubation time in mosquitoes.
Mm-hmm.
And so when the temperatures go down, it doesn't matter the number of mosquitoes, but the time for the virus to grow within the mosquito gets so long that mosquito dies first. So that's
Oh.
how was our hypothesis. That's how colder temperatures drive dengue down. Not because
Mm-hmm.
there's less mosquitoes, but they're living. Their metabolism is slow, and is too slow for it to complete the cycle of transmission within itself. So yeah, so that's another explanation. You see, because sometimes the easiest or the most immediate or more obvious thing is to think that, okay, if population of mosquitoes grow, then dengue will grow. That's not necessarily a direct consequence. It has to go through something else. So one hypothesis is that hypothesis of the metabolic changes that are driven by temperature, not necessarily the numbers.
Okay, that's an interesting question.
Yeah.
Yeah, indeed. So maybe this relationship between the metabolism and how long the virus takes, it's more important in shaping, right?
Yeah, it is one thing, definitely.
Okay, yeah. Yeah,
Yeah.
that's my impression now that I'm focusing more on, especially on dengue. It's like there's so many ingredients, right?
Yep.
And is it like temperature dependent, the incubation period in the mosquito?
Yes, it is. Yeah, it's
Okay,
been shown
okay.
in the
That's
lab.
interesting.
So it's very easy to really get a grasp of that experimentally, but in the field it should follow pretty closely.
I agree. Okay, well, thank you for the provocation. Davi?
Okay, so another question that I have, did you cross this data? I find it really interesting your idea of somehow we have to measure risk, and the idea of risk can be related to the factors that might like that if you have the disease, then you have a high risk of actually getting a bunch of
Yeah,
cases because you
yeah.
have like the all the stages set for the worst to come. So I find it really interesting the idea of trying to actually to estimate something that is not directly related to the problem at hand. So instead of trying to estimate risk by estimating actually the number of cases, you're trying to estimate something else, which can be really nice because uh, if you're measuring the probability of something actually have the actual variable that you're trying to estimate the risk for, you might be confounding things. But my question is whether you actually, if you, oh, I think you have spoken with your hands. Okay, that's a message
Don't worry.
appeared here. I got confused. It's something about Zoom. So
Mm.
my question is whether you actually saw if there is any correlation between the number of mosquitoes and the dengue incident cases for these places where you collected the data. So do you
Okay.
actually see like higher dengue incidents when you have this higher number of mosquitoes, or is it like not really clear?
Okay, right. So the short answer is no, because for this part of the work, I did not look at the dengue data in these locations. There is some dengue data, but I was not, let's say, in charge of working with it yet. But there's one thing that these locations, dengue is really not endemic. So it's quite different from the regular Brazilian city where we know dengue will be there, right? Every season. And then the abundance of mosquitoes is somehow related to, we believe it's to be somehow related to the risk, right? That's the hypothesis, but like Flavio said, we have many elements. But in these locations, uh, usually the dengue outbreaks are associated to some importation. So you won't have a dengue outbreak every year in Miami, but when you have, it usually can trace back to someone who traveled from an endemic region. And then you can even see some local chains of transmissions. Some outbreaks are really contained, some are a bit bigger. So the question for them, for this type of location, is more about if we import a case today, what is the likelihood that it will generate an outbreak? And that's what the mosquito abundance or the aegypti abundance is telling you for these locations. Then, yeah, if we speak of an endemic location, that's different because we can assume the virus will be there. And then it's more about this relationship that you asked me about. But yeah, the answer is no, I did not look at the dengue data for those locations.
Okay. Does anyone want to do another question? No? Okay. Paulo, thank you very much for the wonderful talk and the wonderful marriage between Inframind and Mosqlimate you are proposing us,
Oh,
we accept you.
oh, great. That's great. Thank you. Thank you so much. It's a honor. Yeah. When I started
Thank you
looking
very much.
up, I was so happy to see that there is a nice platform already developed. So I really like the work you guys did. So thank you so much for the invitation. It's my pleasure.
Thank you very much for the brilliant research.
Thank you.